This lecture is one of the about mitosis, one of the most important processes that a cell can ever do. Mitosis is the replication of the cell's nucleus, and cytokinesis, which happens after mitosis, is the division of the actual cell cytoplasm. The end result would be two cells that are identical to each other. If mitosis did not occur, you would start out as one cell and just stay that way. Mitosis and cytokinesis has to occur to replicate the cells so that we can create multicellular organisms, and even single cells have to do it in order to keep alive on the planet, to replicate themselves. So it's a very, very important and powerful process. In mitosis, or actually in the cell cycle, there are many phases, as you can see in this diagram here. Interphase is the phase that the cell is in when it's not doing mitosis. It's in between mitoses. It's when it's doing its everyday life. It could be a cell in your pancreas, a cell in a leaf of a oak tree. It could be a leaf cell in a blade of grass. It's just doing its job as a cell, a living cell on the planet. That's interphase. Now, G1 is exactly that. G1 of interphase is when the cell is doing its job. Many cells stay in G1 for years. And then eventually they may get a signal to start replicating process. The replicating process begins in interphase S. Interphase S is when the DNA of the nucleus actually replicates itself and creates two new DNA molecules. Identical to each other. However, the DNA is wrapped in the nucleus like a great big complex ball of yarn. So when the DNA replicates, the copies of DNA are all tangled in with each other, and it's very, very difficult for them to separate. So the cell has to devise a way in order to do that, and that's the function of mitosis. When it gets into G2, G2 is just getting things ready for mitosis. It's starting the breakdown of the nucleus, making enzymes and structures that are required for mitosis to occur. And when that happens, we start the mitotic process. Now, this is a cells that are actually in interphase. And you can see that they're just ordinary nuclei. They're doing their thing. These are probably plant cells. And in some of them, you could actually see a nucleolus within the nucleus. The nuclear membrane is intact, and the DNA is not visible because it's in little wispy strands called chromatin. And the chromatin is just not it's so thin and small and wrapped around inside the nucleus like yarn that it's hard to see. The genes though are doing their job, and they're doing the function of the cell. Now, within interphase S, you see what's happening here is that the DNA, the double helix, is actually opening up. And as it opens up, each side of the DNA is making a complementary copy of itself. And when this is all done, you're going to end up with two exact copies of the DNA molecule. This is happening during interphase S DNA synthesis. Here is a diagram, or actually a photograph in an electron microscope of 3% of one chromosome. And you can see that the DNA is pretty well tangled up, and how much DNA there actually is in there. If you can imagine this DNA actually copying itself and making two copies of this whole thing, and then having to separate the copies, that would be a very daunting task. And that's what mitosis's job is, to separate the copies of the DNA so that you can create two identical nuclei. Now, the way it does that is pretty ingenious. What happens is that the DNA, which looks like the previous diagram, or actually photograph, is coils down. It coils down kind of like a rubber band does. You just keep coiling it and coiling it and coiling it and coiling it until instead of being long and thin, it becomes short and fat and a little lumpy. And if you had two rubber bands that are wrapped into each other and they replicated, and you tried to separate them, they'd be tangled. But if you can coil them down into small little lumps, now you could easily separate the replicates. And that's the way, the trick, in a sense, that mitosis uses in order to be able to separate the DNA copies. They call that DNA condensation. In this diagram, this photograph actually on the left, you see a completely condensed chromosome, and it looks like it has two halves held together by a sort of a belt in the middle. Each one of those DNA copies there are exact copies of each other, and the little belt in the middle there is called a centromere or a kinetochore, and it's literally holding the two copies of the DNA together before they get to separate in mitosis. Here is the phase of mitosis called prophase. Now, in prophase, what's happening is that the DNA is coiling down. The nuclear membrane is breaking down, and the chromosomes actually exist now as two identical DNA strands that are all coiled down. The DNA strands at this point are called chromatids, held together by that centromere. And it's a double chromosome because there's two DNA molecules that are exact copies of each other stuck together. That's prophase. And here we have metaphase and anaphase. And in metaphase, there is a spindle formed. A spindle is made of microtubules, and you can barely see it in this diagram, or actually this photograph. In metaphase, the chromosomes line up at the middle or the equator of that spindle, and they look like a little, sort of they're lined up like little soldiers there, all lined up in a row, and they're actually attached to the center or the equator of that diamond-shaped spindle. The spindle, again, are microtubules, and each chromosome is attached to one of the microtubules on the spindle. And that's metaphase when they're all lined up like that. In anaphase, you see that the chromatids are now separating. They're now individual chromosomes, and they're moving down the spindle. Actually, the spindle is pulling them apart. And the centromere splits, the kinetochore is attached to the spindle, and the spindle is literally pulling these double chromosomes apart into single ones. And they're going to migrate down that spindle to the each end of it, called the poles. And the poles will eventually have one set of chromosomes, and then the other pole would have another set of chromosomes on the other side of the spindle. Here we have telophase or telophase. And what's happening here is that the nuclear membranes are forming around the newly separated DNA strands, and they're uncoiling. It's kind of like the rubber bands are uncoiling, and they're becoming that wispy chromatin again, and starting to do their job as controlling and being the program of the cell, and getting back to normal. In the diagram here, you also see that the cell is pinching in, and that is cytokinesis. Cytokinesis is when the cell cytoplasm actually divides into two cells once the two nuclei are formed in each one of the two new cells. And here's a diagram of the whole thing in one shot. And you can, if you take a few minutes and just look at that, you'll see what the chromosomes are doing. And in G1, that's just ordinary life. A cell can live that way for years, doing its thing. When it gets a signal to replicate, it goes into interphase S. In interphase S, you can see the DNA is doubled. It has copied itself. And so now it's a tangled up mass in there of two sets of DNA, which are now, hard to separate. When you get to G2, you see that those little centrioles are starting to form the spindle, and they're moving to either side of the cell. The centrioles are little structures that create microtubules, and they form the spindle of the in the cell for the chromosomes to move on. And then you get to prophase, and when you get to prophase, you see that chromosomes are already coiled down, easily now separated from each other, and they're all double chromosomes because the DNA replicated. Each one of those is an exact copy of the one that's attached to. And the little dot in between is the centromere or kinetochore that are holding the identical chromatids together in the double chromosome. You notice also the nuclear membrane is starting to break down in prophase. When you get to metaphase, you see there's no more nucleus. There's your spindle. It looks like a little diamond, and on either end of the spindle is a centriole. or sometimes called a centrosome, depending on what kind of an organism it is. And then you have the chromosomes kind of lined up smack in the middle of that spindle or the equator, attached by the kinetic cords to the spindle. Once they're lined up like that, then the spindle fibers actually pull the chromosomes apart. Centromere splits and the chromosomes move to either end of the spindle, either pole of the spindle. And they migrate down there. It takes only a minute or less. And then you get to telophase. And in telophase, you see that the two new nuclear membrane are forming. The DNA is uncoiling, getting back to normal DNA or chromatin again, getting back to controlling the cell. And you also notice that the little line in between there is an indication of cytokinesis, where the cell cytoplasm is actually dividing to form two new cells. Here is a nice setup of photographs of a whitefish embryo, and you can see each phase very, very clearly here. Just take a look at them and see if you can, identify what's going on in each one of those phases. And we get one here. This is a plant root. It's actually an onion root. And if you look around, you'll see some phases. I see a metaphase, couple of metaphases, as prophases. A lot of cells are in interphase. There's anaphase, and I don't know if I see a telophase anywhere, but there might be one. But this is an onion root cell slide that really shows, the way the chromosomes look like when they're condensed down and in the various phases. This is a cell doing cytokinesis. The cytoplasm is dividing into two new cells. Nuclei are already formed on either end. There's a nucleus on one and a nucleus on the other. Mitosis is complete. Cytokinesis occurs. Now this is a cancer cell. Cancer is mitosis that is not controlled. And mitosis, because it replicates the cells and makes new cells, is extremely powerful. If it keeps doing it without being controlled, you can create a lump of cells that are inside your body and out of control, growing bigger. That lump would be called a tumor. And of course, tumors cause cancer. And so cancer is actually caused by cells that can't control their mitosis, and they just keep growing into big lumps of cells inside your body. And in breast cancer, for example, you see a tumor or a lump of cells forming inside the breast. And because the cells are replicating, the tumor gets bigger and bigger and bigger, and it's out of control, gets really, really big, and it ends up, getting some of the cells into the lymphatic system, and then the cells can metastasize, which means that they start to move throughout your whole body through the bloodstream and the lymph stream, and really cause a serious cancer. Now, that's about it for mitosis. You can review this at any time, and there's a quiz coming up.